drug discovery process, especially in the target-based approach, quantifying target engagement is essential for building structure-activity relationships and developing clinical candidates. It also helps to describe drugs' mechanism of action by allowing scientists to understand the relationship between drug-target binding and efficacy. Measuring whether a compound interacts with its intended target is extremely important, as skipping target engagement can lead to improper scientific conclusions, wasted time and resources, and ultimately to a compound that fails later in drug development pipeline. During drug discovery projects we ran for our customers, we used different methods to show or to prove that our compounds were really binding to the target of interest. Of course, we started from basic and usual biophysical technologies such as SPR, TRIC, ITC or FP. So basically the methods that identify binding events by measuring refractive index shifts, energy transfer, heat release and absorption, and changes in fluorescence. But the problem was that those technologies don't really tell you anything about what is really happening inside cells, whether the compound actually binds the target of interest in a real cellular environment, so in the presence of the whole complexity that can influence its behavior. That's where NanoBRET became our method of choice. But we soon realized that we didn't have any orthogonal or confirmatory technology to double-check results generated by NanoBRET. And that's how we came across Medicines Discovery Catapult and their CPSA, Chemical Protein Stability Assay.
The detection of increased protein stabilization in the presence of ligand under denaturing conditions is a well-established technique that can be used to measure target engagement. This is the principle for several different commercially available technologies. The need for large amounts of purified protein for some techniques, or a lack of affordable access to others, meant an alternative approach needed to be sought. Though utilised previously, assays using chemical denaturants can be low throughput, complex, and incompatible with some detection technologies. We felt that this was an area of assay development that could be investigated and optimised to generate more successful data than seen previously, while also generating a format more compatible with screening approaches. Our main design goals for the development of a suitable target engagement assay were that it should be simple to run to allow access for any research to use it, a single well format to ensure greater reproducibility, scalable to allow use for compound screening, and also cost-effective. MDC has developed a proprietary lysate-based chemical protein denaturation technology to confirm cellular protein target engagement by monitoring compound binding-induced stabilization in a simple, automation-friendly, scalable, cost-effective assay. The Chemical Protein Stability Assay, or CPSA, is a simplified, single-well, plate-based workflow without plate transfers, suited to compound screening and hit finding at up to 1536 well capacity, and is compatible with multiple assay endpoints. The patented technology is not dependent on the labelling of a tracer ligand or modification of the target protein. The use of cell lysate fits well with large-scale applications where confounding cellular permeability effects are not desirable at early hit ID, while also avoiding issues with large-scale purification of protein targets. The simplicity of the workflow and reagents reduces assay variability and produces consistent data. CPSA is broadly applicable to a wide range of cellular targets.
To understand how the assay works, we have to consider the underlying principle. Proteins, when exposed to stress, tend to lose their secondary and ternary structures and form large insoluble aggregates. The protein structural stability can be estimated from quantification of the remaining soluble protein. Compound binding to a protein will stabilize its structure, and therefore the expectation is that it will increase the fraction of remaining soluble protein. So far, the most commonly used stressor has been temperature. The approach is in principle valid, However, temperature is a factor that is very difficult to control in a precise manner in high-throughput applications. The discovery that proteins can be denatured using organic solvents has been the breakthrough that enabled miniaturization and upscaling target engagement assays.
To put together a compelling case for the use of CPSA over other target engagement assays, a package of validation data was created. CPSA was run head-to-head with an alternative thermal shift assay using p38-alpha MAP kinase as a test protein of interest. As a well-characterized target with a wide range of commercially available inhibitors, we were able to run parallel assays using the same compound samples and protein starting material. Using 23 different compounds with a range of potencies, target engagement to p38-alpha was assessed in both assays by running in the same 384-well concentration response format. This generated a compelling set of data that shows a high correlation between the two techniques, with an R value of 0.79. The reality of this is that the CPSA can generate data that is directly comparable to existing technologies, but uses a simpler workflow in a more cost-effective assay.
At Solvita, we have transferred and independently validated CPSA assay. From the onset, the method proved easy to implement, reproducible, and it generated high-quality data. As opposed to previously used thermal denaturation methods, CPSA allows to distribute the stress very uniformly across the multiple plate. That means that the internal variation of the data is low, and it can be used with a 384-well plates and even with a higher density plates. After initial transfer and validation, we decided to test the suitability of CPSA to other target classes. We have selected BTK and KRAS. Those two proteins are immensely important in modern oncology. They are mutated in many cancers and are attractive drug targets. CPSA was able to identify drugs that specifically bind the mutated but not the wild-type version of KRAS. With BTK, we applied the method to a different class of inhibitors, covalent binders. These are the inhibitors that irreversibly bind the enzyme and inhibit its activity. And also in this case, CPSA proved to be an effective tool to demonstrate the binding of a covalent binder to its intended protein target. CPSA is now fully integrated into our target engagement platform. NanoBRET is great for living cells, however, it requires making specific genetic constructs and also fluorescent tracers for competitive binding, while CPSA is a label-free method and it removes this requirement. Therefore, it is very applicable at the early stages of drug discovery, where speed and agility matter most. By reducing the technological and financial barriers to high-quality target engagement data, CPSA makes the whole drug discovery process faster, more effective, and more accessible to all innovators.
As the first CRO to license CPSA from MDC, we see this collaboration as a clear example of how shared innovation can accelerate the scientific progress. By bringing CPSA into the project workflow, we translate MDC's innovation into robust client-ready applications that can support faster and better decision-making in early drug discovery.